vaccines are developed, as well as the history that led to their creation and refinement. The journey to understanding vaccines is one that also reveals the incredible intricacy of the human body and the complex relationship between humankind and infectious disease. The immune system is a highly sophisticated network within our bodies that protects us from infections, constantly identifying, targeting, and eliminating potentially harmful invaders. The immune system is incredibly nuanced, capable of distinguishing between billions of molecular shapes to recognize pathogens and mount a tailored response. The immune system is broadly divided into two main parts. There's the innate immune system, which serves as a general first line of defense, and then there's the adaptive immune system, which provides a specific response to particular pathogens. This distinction is key to understanding how vaccines work, as vaccines leverage both aspects of the immune response to train the body against certain diseases. Now, the innate immune system is the body's immediate response to foreign invaders. It's rapid, typically taking place within minutes to hours of infection, and it's broadly effective against a wide range of pathogens. When pathogens breach physical barriers like the skin or mucous membranes, they encounter cells of the innate immune system, which include phagocytes like macrophages and neutrophils, natural killer cells, and dendritic cells. Macrophages are large cells that eat pathogens by engulfing them and breaking them down. Neutrophils, which are among the most abundant white blood cells, rush to the site of infection to destroy pathogens with toxic chemicals. Natural killer cells play a particularly important role against viruses and cancerous cells, detecting and eliminating infected cells to prevent the spread of infection. Innate immunity is also supported by various physical and chemical barriers, such as the skin, stomach acid, and antimicrobial peptides. When a pathogen is detected, these barriers work in concert with immune cells to block entry or to destroy the intruder. Now, despite the innate immune system's importance, it can't specifically remember pathogens that it encounters, and it's incapable of distinguishing between different strains of a pathogen. For example, it might attack two different bacteria with the same strength and approach. This is why the adaptive immune system, which provides tailored and long-lasting protection, is also essential. The adaptive immune system, or acquired immunity, is slower to respond initially, but it offers a targeted and highly specific response to pathogens. It involves specialized cells called lymphocytes, including B cells and T cells, which recognize and remember specific pathogens. The adaptive immune response is tailored to individual pathogens, meaning that it can distinguish between different types of bacteria and viruses, and even between strains of the same pathogen. When exposed to a pathogen, the adaptive immune system takes time to recognize and respond, often taking days to activate fully. But once it's recognize and bind to specific molecules on the surface of pathogens called antigens. Antibodies neutralize pathogens directly by binding to them and preventing them from infecting cells. They can also tag pathogens for destruction by other immune cells. Each B cell has receptors on its surface that match a specific antigen, like a lock and a key. When a B cell encounters an antigen that fits its receptor, it becomes activated, proliferating and producing large quantities of antibodies tailored to that specific antigen. T cells, on the other hand, have two main types. They're helper T cells and cytotoxic T cells. Helper T cells coordinate the immune response by signaling other cells and releasing cytokines, which are chemical messengers that direct the activity of other immune cells. Cytotoxic T cells, also known as killer T cells, target and destroy infected cells, helping to prevent the pathogen from spreading. An extraordinary aspect of the adaptive immune system is its capacity for immunological memory. Once an infection is cleared, a subset of B cells and T cells become memory cells, which persist in the body and are ready to respond more quickly and effectively if the same pathogen is encountered again. This process allows the immune system to mount a faster and more efficient response upon reinfection, often preventing the disease from developing or at least significantly reducing its severity. Immunological memory is the principle that vaccines utilize to protect against disease. Vaccines work by simulating an infection in the body, prompting the immune system to produce a response without actually causing illness. This trains the immune system to recognize the pathogen if it encounters it in the future, allowing it to respond swiftly and effectively. Vaccines expose the immune system to specific antigens, molecules from pathogens that the immune system can recognize and remember. By doing so, they prepare the immune system to fight off the real pathogen if it ever encounters it in the body. Vaccines come in different forms, depending on how the antigens are presented to the immune system, and each type has unique characteristics and benefits. Now, one of the earliest types of vaccines is the inactivated vaccine, which contains pathogens that have been killed or inactivated. Now, these pathogens can't cause disease because they're no longer alive, but their antigens are still intact, allowing the immune system to recognize them and mount a response. The inactivated polio vaccine is a classic example. Inactivated vaccines are generally very safe because the pathogen isn't alive, so it can't replicate, it can't cause disease. But because they don't replicate in the body, they may require multiple doses or booster shots to provide lasting immunity, as the immune response to inactivated pathogens can be much weaker than to live pathogens. Now, live attenuated vaccines contain weakened versions of pathogens that are still alive, but less virulent. These weakened pathogens are designed to replicate in the body without causing serious disease in healthy individuals. Because they mimic a natural infection, live attenuated vaccines tend to stimulate a strong and lasting immune response. The measles, mumps, specific parts of the pathogen are used, usually proteins or sugars that trigger an immune response. By isolating these components, scientists can create vaccines that are highly specific to certain pathogens and have minimal side effects. For example, the hepatitis B vaccine contains only a piece of the virus, which is enough to stimulate a protective immune response. The human papillomavirus, or HPV vaccine, is another example, providing protection against the virus that causes most cervical cancers. Because these vaccines don't contain live pathogens, they're safe for people with compromised immune systems, but they may also require multiple doses to ensure a lasting immune response. Now, a newer type of vaccine, the toxoid vaccine, is specifically designed to protect against bacteria that produce toxins, which are harmful substances that can cause illness even in the absence of bacterial infection. Toxoid vaccines contain inactivated toxoids, that can stimulate an immune response without causing harm. The tetanus and diphtheria vaccines are examples of toxoid vaccines, as these diseases are caused by bacterial toxins rather than by the bacteria themselves. By targeting the toxin rather than the bacteria, toxoid vaccines provide protection against the disease. Another really fascinating development in vaccine technology is the advent of mRNA vaccines. These vaccines use a snippet of genetic material, a messenger RNA or mRNA, from the pathogen rather than the pathogen itself. When administered, the mRNA enters cells and instructs them to produce a protein that mimics one found on the pathogen. The immune system recognizes this protein as foreign, mounting a response and crafting memory cells without exposure to the pathogen. mRNA vaccines are highly versatile, and they allow for rapid design and production, making them a fairly promising platform for future vaccine development. DNA vaccines represent a similar approach, but they use DNA rather than mRNA to instruct cells to produce a protein. DNA vaccines are still largely experimental, but they have shown potential for various infectious diseases. Now, they work by introducing a piece of DNA from the pathogen into the body, which cells then use to produce an antigen. This antigen triggers an immune response, providing protection against the disease. Now, DNA vaccines are stable and relatively easy to produce, but they face technical challenges in ensuring efficient delivery to cells. The development of vaccines is a lengthy and complex process requiring extensive testing to ensure that they're safe and effective. Vaccine research begins in laboratories where scientists study pathogens to identify antigens that might trigger a protective immune response. After antigens are identified in a lab, the path to a viable vaccine involves rigorous testing, refinement, and collaboration between scientists, regulatory agencies, and public health experts. A vaccine begins with preclinical studies where researchers tested in cell cultures and animal models to assess its potential for safety and effectiveness. This phase is crucial for identifying any initial adverse reactions and determining if the vaccine triggers the desired immune response. public health organizations, governments, and international bodies work together to ensure vaccines reach everyone who needs them, often establishing partnerships with local health organizations to facilitate distribution. Immunization programs, a core component of public health, are built on the success of vaccine distribution. These programs aim to ensure that as many people as possible Understanding pathogen evolution is also vital in vaccine development, as pathogens can change over time, potentially reducing vaccine efficacy. This phenomenon, known as antigenic drift, occurs in diseases like influenza, where the virus mutates frequently, requiring annual updates to the vaccine formulation. Researchers study these mutations closely to predict future strains and design vaccines that provide optimal protection. Similarly, diseases caused by bacteria with complex life cycles, such as malaria, require innovative vaccine approaches, as traditional methods may not offer lasting immunity. Looking forward, researchers are also exploring vaccines for non-infectious diseases such as cancer. Cancer vaccines aim to stimulate the immune response to target and destroy cancer cells, which are often challenging to distinguish from healthy cells. Some experimental cancer vaccines are designed to enhance the immune response to specific tumor-associated antigens, potentially leading to new treatment options. Vaccine technology is even being explored for autoimmune diseases, allergies, and other conditions, as scientists continue to harness the immune system's power to improve health beyond infectious disease prevention. Environmental factors also play a role in the spread of diseases and the effectiveness of the vaccine. Climate change, urbanization, and global travel have increased the risk of disease outbreaks, making vaccines more critical than ever in preventing the spread of infectious diseases. Warmer climates and disrupted ecosystems